Okay, welcome everybody. This is Chapter 10, the discussion on the circulatory system. We'll be starting today with a discussion of blood. This is the one of two really fluid tissues in the human body. The other is lymph. It's classified as connective tissue because it has very few cells compared to the amount of extracellular component that's in the blood, and in the blood that's called the plasma. It's made up of living cells, which we call the formed elements, and the non-living matrix, which of course is called the blood plasma. Now, if we were to take whole blood, if we were to collect a sample from you and centrifuge it, what we would find after we spun it in the centrifuge is that the denser components of the blood would sink to the bottom and form a packed layer of red cells, and this would represent something that we call the hematocrit. It's the percentage of your whole blood volume that's represented by your erythrocytes. And on average, that's about 45% of your blood volume. The buffy coat would be a layer on top of the packed red layer, and it contains the white blood cells called the leukocytes and the platelets called the thrombocytes, and they aid in blood clotting, and they make up less than 1% of the blood. The buffy coat is a thin whitish layer between the erythrocytes and the plasma, and the plasma makes up basically the remaining 54% of blood if you count the 1% that the buffy coat takes up. Your blood's hematocrit will change depending on the demand of tissues for oxygen. Under high oxygen demand conditions, the hematocrit will rise, and under low oxygen demand, the hematocrit will stay right around that 45% mark. And so this is just a chart showing you the different components of blood. You can see that most of it really is water. This is the plasma component, the 55%. And then you find a lot of electrolytes in blood as well, things like sodium and potassium, calcium, magnesium, chloride, and bicarbonate. And what these components do is, for instance, calcium is important in building strong bones and also in muscle contraction and nerve transmission. Sodium and potassium are critical in maintaining membrane potential, which is a way that living cells store energy, kind of like a battery, through the separation of charge. It's provided primarily by the sodium-potassium pump. Magnesium is a major component of enzymes. It's also a building block of DNA. It's also important in biosynthetic processes in the body, and it's also critical in the production of ATP, in that it participates in components inside the cell that produce ATP in the mitochondria. The bicarbonate and the chloride are critical for maintaining the pH balance of the blood. We have to keep the pH close to 7. If it gets too high or too low, what will happen is that membrane potential will be changed, and the result will be a shutdown in electrical activity, which will shut down the muscular system and the nervous system. It also can result in the denaturing of proteins, and that will cause chemical reactions that they help happen to cease. Remember that enzymes basically are facilitators of chemical reactions, in that they allow the reactants to collide with each other in a way that will always produce a product. The plasma proteins include albumin, which is an important component of the plasma that maintains its volume, so it's important in osmolarity of blood plasma. Fibrinogen, which is critical in the clotting cascade. Clotting is one of the ways that we keep from bleeding to death in the event that we're injured as a result of a puncture or a laceration. And then globulins, which are there primarily as defense molecules. What they do is to bind to specific molecules on the surfaces of pathogens called antigens and tag them for destruction by the immune system. And also These proteins play a role in buffering. Substances that are also transported by the blood include nutrients, waste products, respiratory gases, primarily oxygen and carbon dioxide, and hormones, which are chemical signals that are produced by a variety of glands throughout the body that serve to keep the body in homeostasis. The way that they travel, again, is by the beating action of the heart, and the way that they recognize their appropriate tissue is by binding to the appropriate receptor. Now, if we look at the formed elements of blood, we find that most of them are erythrocytes, and these are our oxygen and carbon dioxide carriers. You got about 4 to 6 million of them per cubic millimeter of blood. Next behind them are the leukocytes, and these are the white blood cells. Unlike the erythrocytes, these have a nucleus, and they're involved with primarily defending your body against disease, either in a nonspecific or a specific fashion. You've got between 5 and 10,000 of these per cubic millimeter of blood. They come in two broad categories: the granulocytes, which are the neutrophils, the eosinophils, and the basophils, and the agranulocytes, which are the lymphocytes, and the monocytes, which eventually become macrophages. And we'll talk, when we get to the chapter on the immune system, about exactly how it is that these cells keep you disease-free. But essentially, if we were to boil it down in a nutshell, they use a variety of chemical weapons in order to neutralize the pathogen before it gets a chance to make you sick. The granulocytes shown here are primarily involved in what we know as our innate immunity, whereas the agranulocytes are involved in our adaptive immunity. Okay, they are what recognize a specific type of pathogen and go out and destroy it, and then in the course of facilitating that attack, they will generate memory cells, which are designed to keep a record of the method used to destroy that particular pathogen, and should that pathogen show up again, the lymphocytes will then generate an army of cells that will take out that pathogen before it gets really a chance to make you sick. The platelets are essentially fragments of cells called megakaryocytes. They do not have a nucleus, and basically they are membrane-enclosed sacs of clotting factors. Clotting is an important process in hemostasis. This keeps us from bleeding out in the event that we're injured, so that we don't hemorrhage to death. If we look at the physical characteristics of blood, it has a range of colors. Oxygen-rich blood is scarlet red, whereas oxygen-poor blood is a dull red, which appears blue when you look at it through the white coating of the veins that carry the blood back to the heart in the systemic circulation. The pH has to remain between 7.35 and 7.45. Again, if we get too far outside that range, enzymatic reactions will shut down, membrane potential will cease or be changed significantly, and that can result in death. Blood temperature is slightly higher than body temperature at about 100.4 degrees Fahrenheit. In a healthy man, the blood volume is about 6 quarts, and blood makes up about 8% of your total body weight. Plasma is composed of about 90% water, and it includes nutrients, electrolytes in the form of dissolved salts, respiratory gases, hormones, which are chemical signals carried throughout the body to maintain homeostasis, secreted by the endocrine system, and then waste products, which are generated as a result of normal metabolism, and then also plasma proteins, which help maintain the osmolarity of blood and also regulate blood clotting, as well as defense mechanisms against disease. So it's important that we maintain circulation, because without these components, particularly the nutrients and the gases, the cells would quickly die. The cells need a constant supply of nutrients and oxygen in order to make enough ATP in order to live. Plasma proteins are the most abundant solutes in the plasma. Most plasma proteins are made by the liver. Various plasma proteins include albumin, which regulates osmotic pressure, and clotting proteins that help keep us from losing blood in the event that we're injured. Otherwise, we would bleed out, and the result is that the cells would lose the access to the oxygen and the nutrients they need to make ATP in order for them to continue to live. The antibodies help protect the body from pathogens, and what they are sort of like magic bullets. They bind to a specific collection of molecules on the surface of a pathogen called an antigen, which is kind of like an ID tag that indicates to the body that you have something inside you that doesn't belong there. And essentially what the antibody will do when it binds to the antigen is it will tag that particular pathogen for destruction, and you'll remain disease-free. And in the course of doing that, the cells that produce the antibodies, which are called B lymphocytes, will generate copies of themselves called memory cells that will stay in your body for decades. And should that same pathogen come by a second time, what will happen is that because of this large standing army of B and T lymphocytes that were generated during the course of the attack on your body's system, we'll be able to quickly neutralize the pathogen before it gets a chance to produce disease. And that's a phenomenon in the immune system that we call memory. Okay, the blood plasma is the extracellular matrix of blood. Acidosis is the condition that results when the pH of the blood drops too low. Again, this will change membrane potential, and that will result in malfunctioning of the nervous system and the muscular system, and it can also eventually lead to protein denaturation and the cessation of chemical reactions that keep the cell going. Alkalosis is the condition where the blood becomes too basic. In each scenario, the respiratory system and the kidneys try to restore blood pH to normal by adjusting the amount of carbon dioxide in your system and by adjusting the amount of bicarbonate and the amount of protons that are in the body fluids, in the plasma and in the tissue fluid. And they do this by changing, in the kidneys' case, what they secrete, and in the respiratory system by changing the rate and the depth of respiration. So if we look at the formed elements, you can see an example of a blood smear here. The erythrocytes, which are the red blood cells, are the oxygen and carbon dioxide carriers, and you can see them here. They look like pale brown biconcave discs, and the reason for their shape is to help make it easier for gases to move in and out of these cells. The primary protein inside these cells is hemoglobin, which has the unique ability to bind oxygen and carry it from the tissue in the lungs to the rest of the tissues in the body, where it will unload it and simultaneously pick up carbon dioxide for transport eventually back to the respiratory system, where carbon dioxide will be exhaled. The leukocytes are the white blood cells, and you can see them here. They stain sort of with a dark purple nucleus. They come in various sizes and shapes, and they're primarily there to defend your body against disease. And then you can see the platelets here, these little tiny purple fragments, and they facilitate blood clotting. Remember that when we're injured and we bleed, one of the things that we don't want to happen is for us to lose a significant amount of blood, because then the living tissues will be deprived of oxygen and nutrients. The erythrocytes' main function is to carry oxygen. Erythrocytes are biconcave discs, meaning that they have on their top and bottom surface a divot in them, okay? And the ridges tend to be raised, and again that increases surface area, and that allows easy movement of gas into and out of the red blood cell. Their primary protein is hemoglobin. They don't have a nucleus. They lose that during the course of hematopoiesis, which is where we produce red blood cells from the cells that live in the red bone marrow. There's about 5 million red blood cells per cubic millimeter of blood. There are very few organelles in a red blood cell because we want most of the space to be taken up by hemoglobin, which is the workhorse molecule that carries oxygen around the body. Another very important protein that's also inside red blood cells is something called carbonic anhydrase, which is an enzyme that generates a molecule called carbonic acid, and this is critical in maintaining blood pH. It facilitates the combination of water and CO2 to make carbonic acid, which can then dissociate into free protons and bicarbonate, and those two species can drastically change the pH of the fluid compartment that they're in. Now, hemoglobin is an iron-containing protein. It's made up of 4 protein chains, 2 alphas and 2 betas. An unusual-looking organic molecule called a heme, which looks a bit like a spider web if you were to draw it. And in the middle of this spider web sits an iron atom, and it's the iron atom that directly binds to the oxygen molecule. It can bind to it reversibly, meaning that it can let go of the oxygen or hang on to it, depending on the conditions around the hemoglobin molecule. Each hemoglobin molecule has 4 oxygen binding sites. It has 4 hemes, 4 iron atoms, and can bind up to 4 oxygens. And every erythrocyte has about 250 million hemoglobin molecules. Normal blood has about 12 to 18 grams of hemoglobin per 100 milliliters of blood, and that is essentially a direct measurement of the oxygen carrying capacity of your whole blood. The more hemoglobin you've got, the more red blood cells you have. The more red blood cells you have, the more oxygen carrying capacity you have in the blood. Now, looking here at our first formed element, this is the erythrocyte, and the normal amino acid sequence of hemoglobin is ideal for it to bind to oxygen at concentrations that you find in the lung tissue, and for it to release oxygen at concentrations that you find in the tissues in your body that are actively metabolizing. So one of the things that we need to understand about hemoglobin is that it tends to pick up oxygen in an oxygen-rich environment, and it tends to release oxygen in an oxygen-poor environment. It also tends to release oxygen under high carbon dioxide concentrations and under low pHs, and those are exactly the same kind of conditions that you find in active tissue. In the lungs, it's reversed. The pH tends to be close to neutral. The oxygen content tends to be rather high, and the carbon dioxide content tends to be rather low. And so what happens in the lungs is that the hemoglobin tends to let go of the carbon dioxide that it's bound to accept the oxygen that's in the air that you inhale, and that loads up the blood with oxygen so that it can then drop it off to the tissues in your body in order for them to continue to make ATP and to live. But occasionally what can happen is that we can have a mistake in the amino acid sequence of the hemoglobin molecule that is inherited. Such a disease is called sickle cell anemia. And what happens in individuals that have 2 copies of this defective hemoglobin gene is that under low oxygen concentration, the hemoglobin molecules inside the red blood cells will form a polymer, and that will cause the shape of the erythrocyte to change from a disc shape to a sickle shape, looking a little bit like a blade. And the result is that less oxygen will bind to the hemoglobin, due in part to its change in shape. The result of these unusually shaped cells is that they will block capillary beds, and they will choke off oxygen supply to downstream tissue, as well as not binding oxygen effectively themselves. And so the, the cure is to always have these individuals in a normal to high oxygen environment in order for the hemoglobin to stay well saturated and to deliver oxygen to tissues as they need it. There's no cure for the disease because this is something that's present when the sperm meets the egg. It's in the DNA. Individuals that have one copy of this defective gene are said to have what we call sickle cell trait, where they carry the defective gene, and they can sometimes have problems with oxygen supply under extremely low oxygen concentrations, but generally um, it's not a problem delivering oxygen to the tissues in people that only have one bad copy of the gene, because there are enough normal erythrocytes to deliver the oxygen effectively to the tissues, even at somewhat low oxygen concentrations. But carriers for this condition can pass it to their children, and if you have two carriers that get married, there is a one-fourth possibility that they're going to have a child with full-blown sickle cell anemia as a result of inheriting two bad copies of the gene. And if you're interested in why that is, I'll diagram that in class for you. Okay, polycythemia is a disorder that results from an excessive or abnormal increase in red blood cell count. It can be caused by bone marrow cancer, where it's called polycythemia vera, and it can be a response to life at high altitudes, where we call it secondary polycythemia. Increased red blood cell count will slow the flow of blood and increase its viscosity, so it makes the blood thicker. We generally see this again in people that perhaps live at high altitudes. This is one of the ways that the body adjusts the oxygen carrying capacity of the blood so that we can continually deliver it to metabolically active tissue. But again, it can also be caused as a result of a pathological condition, as we've indicated here, okay, such as polycythemia vera. Leukocytes are our disease fighters. Leuko means white, cyte means cell, so these are your white blood cells. They defend us against disease, and they are complete cells that have a nucleus and organelle. So they're capable of cell division, they're capable of biosynthesis, and they live a lot longer than a typical erythrocyte does, which lasts only about 2.5 trips through the circulatory system before it has to be recycled. It's able to move in and out of blood vessels through a process called diapedesis, where it can actually change its shape and squeeze through cracks and holes in tissues in order to go from one tissue compartment to the next. This is facilitated by amoeboid motion. Okay, basically the cell will crawl in and around the body towards chemicals that attract it. Okay, and this is an example of what we call positive chemotaxis, a way to attract white blood cells to the site, say, of an infected or a damaged tissue in order to defend against potential infection. So they respond to these chemicals in order to keep you disease-free. You have about 5 to 10,000 leukocytes per cubic millimeter of blood. So when people do DNA tests on blood, it's the DNA extracted from the white blood cells that allows them to determine who the blood belongs to. And remember that the platelets and the erythrocytes do not have DNA because they do not have a nucleus. There can be conditions that lead to abnormal numbers of white blood cells. Leukocytosis is an example. This is a white count above 11,000 white cells per cubic millimeter. It generally indicates an infection. Leukopenia is an abnormally low white count, and it generally can be caused by the use of certain drugs, such as corticosteroids or certain anti-cancer drugs. This is one of the reasons why, in clinical practice, we try to avoid extended use of corticosteroids as a treatment for inflammation, because over time it could suppress the immune system to the point where you become extremely susceptible to infection and disease. Leukemia is where the bone marrow becomes cancerous. and produces excessive numbers of white blood cells. Granulocytes are a class of white blood cells that contain granules, which are colored bodies in the cytoplasm that contain chemicals that are primarily there um, to fight disease. Um, Really, all white blood cells have granules in their cytoplasm. It's just that the granulocytes have granules that will stain with a certain type of dye that renders them either red or blue or pink. They possess nuclei that have many lobes. As a result, they're sometimes referred to as polymorphonuclear leukocytes. Poly means many, morpho means shape, and nuclear means the nucleus. These include neutrophils, eosinophils, and basophils. The agranulocytes lack visible cytoplasmic granules. The nuclei are spherical, oval, or kidney-shaped, and they include lymphocytes and monocytes. Um, the neutrophils, the eosinophils, and the basophils have a very distinctive appearance under the microscope after they're stained. The neutrophils have a multi-lobed nucleus with pink staining granules in the cytoplasm. The eosinophils, a multi-lobed nucleus with red staining granules in the cytoplasm, and the basophils, a multi-lobed nucleus that is obscured by the presence of large numbers of blue staining granules in the cytoplasm. And remember that these granules contain chemicals that are there to help them fight disease. So when a granulocyte comes upon an infected tissue or a pathogen, what it will do is degranulate, meaning it'll cause those granules to fuse with the plasma membrane and release their contents into the outside environment. And this will generally produce a reaction in the target tissue that causes it to die, okay, either as a result of fracturing the cell membrane or by triggering chemical processes inside the infected cell that cause it to commit suicide. So this is a list of the white blood cells from least to most abundant: the neutrophils, the lymphocytes, the monocytes, the eosinophils, and the basophils. And there's a very good mnemonic that'll help you to remember the most to least abundant white blood cells. Never let monkeys eat bananas. Okay, so the neutrophils, the most abundant, the basophils, the least abundant, and so on. All of these cells come from hemocytoblasts, which are found in the bone marrow. They're constantly dividing, either to produce lymphocytes or to produce the granulocytes, um, the monocytes, the platelets, and the erythrocytes. Again, all depending on their interaction with certain chemical factors in different tissue compartments that cause these stem cells to develop along a different path and generate a completely different cell type. So you can see how the influence of these chemicals by binding to the cell membrane can drastically affect the types of white blood cells that these stem cells end up producing. Now, if we look at the different types of granulocytes, the neutrophils have a cytoplasm that contains granules that stains pale pink, and a deep purple nucleus that has between 3 and 7 lobes. They function as phagocytes at active sites of infection. They primarily attack bacterial infections, but they also assist in other types of cleanup. Their numbers generally increase during infection. You have between 3 to 7,000 neutrophils in a cubic millimeter of blood when you're not fighting an infection, and the number goes considerably higher when you are, and make up about 40 to 70% of your white blood cells, depending on whether or not you're battling an infection at that point. Eosinophils contain red, coarse cytoplasmic granules that have a figure 8 or bilobed nucleus that stains bluish red, and their function is to kill parasitic worms and other types of parasites, and they also ingest allergen antibody complexes during allergic reactions, and this can stem what we call hypersensitivity disorders, things like anaphylactic shock and asthma, which are essentially an overreaction of the eosinophil to the antibody that binds to the allergen. You have between 100 and 400 eosinophils in a cubic millimeter of blood. This makes up between 1 and 4% of your white cells. The basophils have sparse but very large blue-purple granules that almost obscure a U or S-shaped nucleus that stains dark blue. These granules are special because they contain a chemical called histamine, which promotes the inflammatory response. And the inflammatory response is essentially when a tissue reddens, it swells up, it increases in temperature, and it hurts. Okay, and this is all a way to bring blood to the area of an injury or an infection. The blood contains the white blood cells, and the white blood cells, of course, will battle the pathogen and do tissue repair. They also contain heparin, which is an anticoagulant, and essentially the way that the heparin operates is that it will keep the tissue fluid in a liquid state while forming a blood clot around the area of the injury, and this allows the white blood cell to do battle with the pathogen or the infected tissue in a confined space where it can't escape, and as it does so, it'll produce byproducts of the battle, which we call pus, okay, which are essentially exudates that contain damaged tissue and white cells and dead pathogen. You have between 20 to 50 basophils in a cubic millimeter of blood, and this makes up around 1% of your circulating white cells. Okay, it's the least abundant granulocyte and the least abundant leukocyte in the blood. Now, lymphocytes are special. They're responsible for what we call our adaptive immunity. They have a cytoplasm that is pale blue with a dark purple-blue nucleus. It functions as part of the immune response. The B lymphocytes generate antibodies, which are similar to magic bullets in that regardless of where they're made, they will always eventually bind to their target, which is an antigen that is different from your own in your body. And then T lymphocytes, which are involved in graft rejection and in fighting tumors and viruses. The T lymphocytes come in three flavors. You've got the helper T lymphocytes that facilitate the immune response by producing chemical signals that promote the proliferation of specific B and T lymphocytes designed to fight a particular strain of pathogen. You've got the cytotoxic T lymphocytes, which actually show up where infected tissue is or where pathogens are and blow holes in the cell membrane or secrete chemicals that cause the infected tissue or the pathogen to commit suicide. And you've got the regulatory T cells that turn the whole process off once the pathogen has been subdued. You've got between 1,500 to 3,000 lymphocytes in a cubic millimeter of blood, and this makes up between 20 and 45% of your white cells. The monocytes will eventually become macrophages, which are critical in coordinating our response against infection because what macrophages do is engulf the pathogen, chop it into little bits, and display those tiny bits on the surface of the cell membrane, and that will alarm the lymphocytes that are able to bind to these antigen and histocompatibility protein complexes effectively will be the ones that will be selected to proliferate and to eventually go out and fight the infection. So without really the macrophages and the other antigen-presenting cells, the lymphocytes would have a hard time selecting from among their billion different specificities which particular B and T lymphocytes would be best at fighting that particular infection. So they definitely assist the adaptive immunity in being effective against disease. They're the largest of the white blood cells. They have a gray-blue cytoplasm and a dark purple nucleus that is often shaped like a kidney, and they eventually become the macrophages as we've discussed. They're important in fighting chronic infection, and you have between 100 and 700 monocytes per cubic millimeter of blood, and this makes up between 4 and 8% of your white cells. Now the platelets are important because they help blood clotting. They're derived from megakaryocytes, which are large multinucleate cells that live in the bone marrow. They're needed for the clotting process. The platelet count ranges from 150 to 400,000 platelets per cubic millimeter of blood. 300,000 is considered a normal number. Any higher than that, then we may have some type of clotting disorder. We may be suffering from thrombocytosis. Okay, if the lump number is low, we call it thrombocytopenia. Hematopoiesis is the process of red and white blood cell formation, and it occurs primarily in the red marrow, which in the adult is found in the rib cage, the clavicle, the scapula, the sternum, and the os coxa, as well as the sacrum and the coccyx and the heads of the femur. The long bones of the adult contain yellow marrow, which is primarily adipose tissue. They're all derived from a common cell called a hemocytoblast. What happens when they're exposed to particular chemicals in different compartments is they begin to differentiate. The lymphoid stem cells will produce lymphocytes, while the myeloid stem cells produce the rest of the formed elements. And again, what determines this is where these lymphoid and myeloid stem cells travel to after they divide off the hemocytoblasts. Okay, what they'll do is they'll wander off to different tissue compartments in the body, and in those tissue compartments will be contained chemicals that will cause them to turn into different types of cells, as you can see in the diagram. Erythrocytes are unable to divide, grow, or synthesize proteins because they have no organelles and they have no nucleus. They wear out in about 120 days, and when they are worn out, they're eliminated by phagocytes in the spleen and the liver. The lost cells are replaced by the division of hemocytoblasts in the red bone marrow. So even though your red cells don't live very long, they're constantly being regenerated because you always have healthy marrow throughout your lifetime. And again, one of the reasons that they are so short-lived is because they cannot engage in any repair, which would require biosynthesis. How do we control erythrocyte production? Well, we do this using a hormone called erythropoietin, which is secreted by the kidneys. They produce the most erythropoietin as a response to reduced oxygen levels in the blood. Homeostasis is maintained by negative feedback from blood oxygen levels. So you can see here under normal oxygen levels we have a typical amount of erythrocytes in the blood, but when oxygen concentration begins to drop due to a lowered red count or a lower amount of hemoglobin in the blood or a reduced amount of oxygen, the kidney and the liver will generate erythropoietin, which will stimulate the red marrow to generate increased numbers of red blood cells. As a result, the oxygen carrying capacity of the blood will go up and we'll retain our normal hematocrit. Individuals that live at high altitudes where their oxygen concentration in the air is much less than at sea level tend to have a condition called polycythemia, which is brought about as a result of the increased amount of erythropoietin in their system that causes their blood to thicken as a result of the increased number of erythrocytes, and as a result increases the oxygen carrying capacity of the blood even at these lower oxygen concentrations. Now, white blood cell production and platelet production is controlled by a variety of hormones called colony stimulating factors as well as interleukins, and this makes the bone marrow generate leukocytes, while thrombopoietin stimulates the production of platelets from megakaryocytes. And again, this all happens beginning in the red bone marrow. Hemostasis is the cessation of blood loss from a break in a blood vessel. It involves three phases: vascular spasm, platelet plug formation, followed finally by coagulation. And all of these are designed to do one thing, which is to stem the loss of blood, which would eventually result in tissue death. So if we look at each step in a front-to-back fashion, we begin here with the vascular spasm, which is brought about by smooth muscle contraction in response to chemicals that are released by damaged tissue. So what will happen here is that the smooth muscle in the middle layer of the blood vessel, called the tunica media, will contract as chemicals are released from the damaged tissue around it. This narrows the blood vessel, and this decreases the amount of blood loss. So that's the first step. The next step is platelet plug formation. The collagen fibers that protrude from the damaged tissue will bind to platelets that are always present in the plasma, causing them to cling to the fibers, and they'll release their contents and attract more platelets. The platelets will then pile up and form a platelet plug. In addition to that, as the platelets rupture, they'll release clotting factors that will promote the formation of a blood clot. And this is what we call coagulation. The injured tissues will release a chemical called tissue factor. PF3, which is a type of phospholipid, will interact with tissue factor as well as other blood protein clotting factors and calcium that's always present in the blood, and this will trigger something called a clotting cascade. Prothrombin activator will convert the prothrombin to thrombin, which is an enzyme, and eventually this will result in the production of a fibrin clot. And so here you can see coagulation in the inset. Essentially, the thrombin acts on the fibrinogen to produce fibrin. The fibrin will then trap red and white blood cells and form an insoluble plug, and this is what we know as a blood clot. Okay, so this keeps the enemy out, and it also is going to begin to form the basis for not only destruction of any pathogens, but also the beginning of what we call tissue repair. Okay, so the fibrin traps these red blood cells, the temporary plug is generated, and then the next step is going to be repair of the tissue underneath it. The blood usually clots within 3 to 6 minutes. If it takes any longer, or if it never happens at all, we may have a condition called hemophilia, where we're lacking one of the proteins in the clotting cascade that eventually leads to the fibrin clot. In those individuals, in order to have normal blood clotting, we have to actually supply the missing factor as an injectable or as an oral. The clot remains as the endothelium begins to regenerate underneath it. After that, the clot is then broken down once the tissue is repaired, because now it's no longer needed. Sometimes clots will happen in places that aren't necessarily sites of tissue damage. A thrombus is a clot in an unbroken blood vessel, and if it lodges in capillary beds in the brain, it can cause a stroke. If it lodges in capillary beds that supply the heart, it can cause a heart attack. One of the ways that a thrombus can form is when blood flow slows down. What that does is that causes a transient increase in the amount of clotting factors in the plasma at a particular location, and that can trigger spontaneous clotting throughout the body. An embolus is a traveling clot. What it'll do is break away and float freely in the bloodstream. Those traveling clots can eventually lodge in capillary beds. If they happen in the brain, they can cause a stroke, and if it happens in the heart, it can result in a heart attack. Thrombocytopenia is a platelet deficiency. In these individuals, normal movement can cause bleeding from small blood vessels that normally would require platelets for clotting. Hemophilia is a hereditary lack of one of these clotting factors, which results in continued bleeding at the site of an injury. And again, the only way to fix hemophilia is to supply the missing factor as an injectable or as an oral. Now, one of the things that we have to be aware of when we talk about blood is this concept of blood type. Now, all blood is the same in the sense that it's designed to carry oxygen and carbon dioxide around the body. It's also a method of transporting electrolytes and nutrients and chemical signals such as hormones, and also helping to eliminate waste products. So functionally, all blood is the same. But the reason that we have this phenomenon called blood types is because different individuals will have different antigens on the surfaces of their red blood cells. A loss of blood can have serious consequences for our overall health. It can result in infarction or tissue death. Loss of 15 to 30% causes weakness, and loss of over 30% will cause shock, which can be fatal. Generally, we engage in a transfusion to replace the missing blood. The transfused blood has to be of the same blood group, or else the antibodies that are present in the recipient's plasma will recognize an antigen that isn't present on its own red blood cells and will cause that incoming blood to agglutinate. And that will be the same as having multiple emboli all over the place, in the sense that these agglutinated red blood cells will block capillary beds and choke off blood flow to downstream tissue that can result in tissue necrosis and death. Blood contains genetically determined proteins. Antigens are substances that identify cells as belonging in the body or not. They can be recognized by the immune system through proteins that are called antibodies, or other proteins that are called T-cell receptors. Blood is typed using antibodies that will cause blood with certain collections of antigens on their surface to clump in a process called agglutination. There are over 30 common red blood cell antigens. The most vigorous transfusion reactions are the ones that are caused by the ABO and RH blood group antigens. And these are two separate clusters of genes. The ABO genes and the RH genes are in different locations in human DNA, and they code for different types of antigen molecules. The ABO blood groups are based on the presence or the absence of two antigens, type A and B. The lack of these antigens is grouped as a type known as O. Okay, and what these are collections of sugars that are on the surfaces of red blood cells. The presence of both antigens is called type AB. The presence of only antigen A on the surface of the red blood cell is termed type A. The presence of only antigen B is termed type B. The lack of both antigens is termed type O. In each case, there are going to be a collection of sugar molecules on the red blood cell surface. The A will have a certain collection of sugars, the B will have a different collection, and the O will have a different collection still. Antibodies are always present from birth on against the type A and B antigens in individuals who have type O blood. In an individual with type A blood, they'll have antibodies against the B antigen, and an individual who has both A and B antigens will lack antibodies to both. There is no antibody against the O antigen. As a result, individuals who have type O blood and have that blood apheresed, to have the plasma removed before transfusion, can serve as universal donors, whereas an AB individual who lacks antibodies against either A or B antigens is termed the universal recipient. And we have a little video that we'll watch at the end of this presentation that'll show you exactly how this agglutination reaction occurs. So when we're looking at the ABO antigens on the red blood cells of different individuals, you can think of them as ID tags that indicate to the immune system whether or not a particular tissue belongs inside your body. If there is an antigen present on a pathogen or on a cell that is not recognized as belonging to your body's own tissue, what will happen is that the immune system will be alerted to its presence, and then we will select an army of lymphocytes specifically designed to go out and destroy anything with that antigen attached. So with regard to the ABO blood groups, you can think of these as a subset of these types of antigens. ABO blood group is coded for by a set of genes that are in a specific location inside human DNA. Individuals that have the gene for the A and the B antigen have blood type AB. They express both the A and B antigen on the surface of their red blood cells. As a result, they are not going to have antibodies against their own antigens, so they will lack antibodies against either the A or the B antigen. There is no corresponding antibody against the O antigen. So an individual who's type AB can receive any other blood type, A, B, AB, or O. So they're termed universal recipients. An individual who's type B blood is going to have the B antigen on the surface of their red blood cells, and they're going to have antibodies against the A antigen. So they would be unable to receive either type A or type AB blood. However, they're perfectly capable of tolerating type B or type O blood. An individual who's type A is going to have the A antigen on the surface of their red blood cells, and they're going to have antibodies against the B antigen. As a result, they're unable to receive either AB or type B blood, but they can receive either A or type O blood. And finally, an individual who is type O is not going to have an antibody against the type O antigen because that antibody simply isn't produced. However, they are going to have antibodies against both the A and the B antigen, and the result is going to be that they can only receive type O blood. Now you might think that this is a dangerous situation to be a type O individual and only be able to receive type O blood, but it turns out that type O blood is the most common blood type in the United States, so it's not as if there's going to be a dwindling supply of that blood type anytime soon. When we collect blood from individuals for donation, one of the things that we do is that we remove the plasma and we resuspend the red blood cells in physiological saline. The result is that when we transfuse a particular blood type into another individual, we only have to concern ourselves with the donor antigens and the recipient's antibodies. And that's why in an individual who's type O, we can donate that blood to any other individual because there is no corresponding antibody that will recognize and attack the O antigen. And so this is why they're termed universal donors. Now what you're seeing here is just a look at some of the different blood groups, the antigens present on each type of cell, and the antibodies that are present in those individuals in their plasma. So you can see that the AB individual has no plasma antibodies against either A or B, and remember that there is no antibody against O. The type B individual has the anti-A antibody. The type A individual has the anti-B antibody, and the type O individual has both the anti-A and the anti-B antibody. And so you're looking here in the last column at the different types of blood that can be donated to these individuals. But before we end our discussion of blood groups, one of the things that we have to address is the fact that there is a second blood group called the RH group, so named because it was first described in rhesus monkeys, and it relates to the presence or the absence of a special type of antigen called an agglutinogen, which is indicated by the capital letter D. An agglutinogen is simply another name for an antigen. In the case of individuals who are RH positive, they're going to have the antigen coded by the D gene on the surface of their red blood cells. In individuals who are RH negative, they're going to lack that antigen. So there's only two blood types with regard to the RH blood groups, either RH positive, which means you have the D antigen, or RH negative, which means that you don't have the D antigens. Okay, so you can see here that most Americans are RH positive. The problem with RH incompatibility is that we have to take note of the fact that in RH negative individuals, they can develop antibody against the RH antigen if they've been exposed to it one time. This can happen as a result of a mismatched blood transfusion, where an Rh negative individual gets Rh positive blood. And it can also happen in the scenario of an Rh negative mother who is pregnant with an Rh positive child. What happens during the first pregnancy is that if an Rh negative mother is harboring an Rh positive child at birth, what will happen as the placenta is torn from the uterus is that the baby's blood and the mother's blood will temporarily mix, and that will result in an exposure on the part of the mother. What will happen in the meantime is that she will begin to build up antibody against the Rh antigen as a result of that exposure. If she becomes pregnant a second time with an Rh positive child, it turns out that the antibody against the Rh antigen is small enough that it can actually cross the placenta and get into the baby's bloodstream and attack the baby's red blood cells and cause them to lyse, and that could end up killing the baby. So for a long time, in order for people to get married here in the United States, what we had to do is subject them to a blood test where we would check for Rh compatibility between the husband and the wife. And if the wife was Rh negative and the husband was Rh positive, you had a 50% possibility in each pregnancy of having an Rh positive child. And the danger, of course, is that after the first pregnancy, you ran a 50% chance of killing the child as a result of this phenomenon of the Rh antibody crossing the placenta and destroying the red blood cells. So what happened over time is that instead of having to worry about the results of these blood tests for people with regard to whether or not they could have children, we developed a compound called RhoGAM. Okay, and don't confuse this with Rogaine. Okay, RhoGAM spelled RHOGAM. And what RhoGAM does is it blocks or disguises the Rh antigen so that the woman's immune system never gets exposed to it. So in the scenario of an Rh negative woman trying to become pregnant by an Rh positive spouse, what we would do is we would administer RhoGAM before, during, and after the pregnancy. And when the baby is being expelled through the birth canal, even though the baby's blood and the mother's blood would mix, it won't constitute an exposure because the RhoGAM will hide the Rh antigen from the woman's immune system. So she can have as many Rh positive children as she wants. Now remember that in the case of two individuals who are both Rh negative having children, we don't have to concern ourselves with this problem because there's no possibility that they can have an Rh positive child. Basically, the only way that you can have an Rh positive child by an Rh negative mother is if you have an Rh positive father. Okay, and generally in the general population, most individuals carry one copy of the Rh positive allele, the D gene, and one copy that is what we would consider Rh negative. And so there's usually a 50% chance on any given birth that you're going to have an Rh positive child. In the case of the mother being Rh positive, we don't have to worry about having an Rh positive baby because she's not going to generate antibodies against her own antigen. And so this problem of Rh incompatibility doesn't come up in that scenario. So the danger occurs only when the mother is Rh negative and the father is Rh positive and the child inherits the Rh factor. The RhoGAM shot can prevent the buildup of anti-Rh antibodies in the mother's blood, and as a result, they can have as many Rh positive children as they want. The mismatch of an Rh negative mother carrying an Rh positive child will cause problems for the unborn child. The first pregnancy usually proceeds without any problem because sufficient antibody won't be built up in order to produce this immune assault on the baby's red blood cells. Is a phenomenon that we call erythroblastosis fetalis. But after the first pregnancy, the immune system has become sensitized. In the second pregnancy, the mother's immune system produces enough antibody, or has produced enough antibody, that they can attach the Rh positive blood that's in the newborn by moving across the placenta, getting into the baby's circulatory system, and then causing those red blood cells to lyse. And again, that can kill the baby very easily. Now, blood typing is one of the ways that we avoid the phenomena of unwanted agglutination in the event of a transplant. In the early days of blood transfusion, before we understood how blood typing worked, what they found was that a certain percentage of blood transfusions would be successful and a certain percentage of blood transfusions would end up killing the recipient. And we didn't really understand why at that point. It was with the discovery of these antigens and the advent of blood typing technology that we were able now to ensure that every transfusion takes place successfully. And the way that we do this is that we mix a potential recipient's blood with antibodies against the A or the B antigen in separate wells. And also we also can check for the presence or the absence of the Rh antigen at the same time. And this will allow us to select the appropriate blood type for that individual. So the blood samples are mixed with the antisera. Coagulation or no coagulation leads to determining the blood type. The type of ABO and Rh factor is usually done at the same time. Cross-matching is where we test for agglutination of the donor's red blood cells by the recipient's serum and vice versa. And so what you're seeing here is an example of some different blood typing reactions using different blood samples. So in this individual, we see agglutination in both the well with the anti-A antibody and the anti-B antibody. And so we would assume from that result that that individual's blood had both the A and the B antigen on its surface, and so that individual's blood type would be AB. In this example, we have no agglutination in the A well and we have agglutination in the B well, so this individual's blood type would be B. And over here, we have agglutination in the A well and none in the B well, so this individual's blood type would be A. And finally, down here, with no agglutination in either well, we can assume that this individual is type O. Now, one of the things that we should address before we end this chapter is to talk a little bit about where blood comes from. When we're still inside our mother's womb, the fetal liver and the spleen are our major blood-forming organs. The bone marrow will take over the formation of blood usually by the 7th month in utero. Fetal hemoglobin is a little bit different from the hemoglobin that's produced after birth because we use a different gene called the fetal hemoglobin gene, which is a little bit better at binding to oxygen than adult hemoglobin is. And the reason for this is so that oxygen can be exchanged between the mother's blood and the baby's blood, partly as a result of the difference in oxygen concentration in the blood that's coming to the placenta from the baby compared to the mother's arterial blood that is perfusing the placenta, but also due to the fact that this fetal hemoglobin is better at binding to oxygen than the mother's hemoglobin is. And so what happens is that while the baby is still in utero, this very effective oxygen binding molecule, fetal hemoglobin, is able to scavenge sufficient oxygen from the maternal blood to provide sufficient oxygen to the baby in order for it to fully develop. Remember that the baby has no source of oxygen other than from the mother's blood supply because it's basically floating inside a fluid-filled sac at that point. The lungs and the respiratory system aren't going to become a significant contributor to the oxygenation of blood until after birth. Physiological jaundice can result in infants whose liver cannot rid the body of hemoglobin breakdown fast enough. And so what will happen is that there will be a yellowing of the skin, which is the result of the buildup of some of these breakdown products, bilirubin, in the skin. You'll also see a yellow cast to the sclera of the eyes. Generally, as the liver continues to develop, however, eventually we're able to effectively recycle the red blood cells from that point on. So it's usually just a temporary condition. We also see this problem in individuals who have compromised liver function. For instance, in individuals who engage in a lot of recreational drug use or excessive consumption of alcohol, what can happen is that you can literally kill liver cells. And when you do that, the liver enzymes that are responsible for breaking down the components of hemoglobin aren't produced at a sufficient enough level to maintain this constant turnover and recycling of these components. And the result is that you'll see this jaundice phenomena take place as these individuals begin to get a yellow cast of their skin into the sclera of their eyes as well. Okay, stay tuned for a brief discussion of the blood and blood typing, and then please rejoin me for our discussion of Chapter 11. Thank you.
Type 2 hypersensitivities involve interactions of antibodies and surface antigens of cells, followed by complement-assisted lysis of these cells. A typical example is mismatched blood transfusions. There are 4 different blood groups, based on types of antigens on the surfaces of the red blood cells. People who are type A have A antigens, and those who are type B have B antigens on the surfaces of their red blood cells. Persons who are type AB have both A and B antigens, and those who are type O have neither A nor B antigens. The serum of people with type A blood contains antibodies against type B antigens, and the serum of people with type B blood contains antibodies against type A antigens. Type AB serum contains neither antibody, and O serum contains antibodies against both A and B antigens. If blood from a person who is type B is transfused into a person who is type A, antibodies present in the type A blood react with the surface antigens on the incoming red blood cells. This leads to complement fixation and lysis of these cells. If blood from a person who is type A is transfused into a person who is type B, antibodies present in the type B blood react with the surface antigens on the incoming red blood cells. This also leads to complement fixation and cell lysis. Persons who are type O lack A and B antigens on the surfaces of their red blood cells and are therefore universal donors. Persons with type AB blood lack antibodies against A or B antigens and are therefore universal recipients.
Blood is responsible for transporting oxygen to the cells and removing carbon dioxide for excretion by the lungs. Blood carries nutrients, ions, water, hormones, and enzymes to all body cells and carries away the waste products. Blood regulates the pH and temperature of the body. Blood is composed of the liquid portion, plasma, and solids called formed elements. Plasma makes up 55% of the total blood volume, 91% which is water. Plasma proteins are albumin, to maintain osmotic pressure and water balance, globulin antibodies to help the immune system, and fibrinogen, an essential component for blood clotting. The formed elements are erythrocytes or red blood cells, leukocytes or white blood cells, and thrombocytes or platelets. Red blood cells have no nucleus, therefore they do not divide and only live 120 days. Red blood cells contain hemoglobin that carries oxygen to all cells of the body and then carries away carbon dioxide. There are 5 types of white blood cells, 3 granular and 2 non-granular. Under the microscope, the granular types appear to have grains. They are neutrophils, eosinophils, basophils. Neutrophils phagocytize or eat up foreign substance and secrete lysosomes that destroy bacteria. Eosinophils produce antihistamines that combat irritants. Basophils are also involved in allergic reactions, secreting heparin, histamines, and serotonin. Monocytes also phagocytize bacteria and dead cells, and when they enter the tissue they are called macrophages. Lymphocytes produce antibodies and are crucial to the body's immune system. Bacteria are being destroyed by neutrophils, lymphocytes, and macrophages. Platelets initiate the chain reaction in clot formation.